is Professor Strazewski from Lyon, who's going to be telling us a number of exciting things from his laboratory. And if we could get the first slide up, that would be wonderful. All right, that's looking good. Wonderful. The floor is yours, sir. We look forward to your presentation.
Good afternoon. Good day, everybody. Thank you very much for your kind introduction, Mark. I'm so happy that I can participate at this wonderful meeting and show you some results that I have. First of all, I hope the screen works well. Yes, Strazewski. I'm Peter Strazewski, Peter Strazewski, Pierre Strazewski, Piotr Strazewski. The good thing about this confusion is that all this is me, and so I'll react to any of those names in the languages that I use. I'm in this community of SupraBio for quite some time. I guess since I've worked on amphiphilic peptidyl RNA, and this was quite a designed molecule, designed for anchoring to membranes, yeah. And ever since, we've got further in the project, and we focus now on the difference between, or not the difference, but the transition, and what we can learn as chemists from the transition of inert matter into the living state of being. So, of course, we all know, or we know a lot about, let's say, much more about one direction than the other, and abiogenesis is, everybody talks about it, or many people, but we don't know much about it from the experimental point of view, do we? So there is this saying that you know, or probably all know, omne vivum ex vivo, all living things come out of the living. It has been first formulated by Lazzaro Spallanzani in the mid-18th century. He was a priest and a biologist and a physiologist, studied in Bologna, and you know, a true academic of his time, who was actually the first to experimentally show fertilization, and that way he could contradict to the opinion, the current opinion, for example, that life can come out of dust. And this had a great influence, and more or less 100 years later was deepened very strongly by Louis Pasteur. And now, 150 or 60 years later, I think we need to restart thinking about it again, because we need to see somehow the origin of it. And so we cannot, I mean, maybe not out of dust, but I think it is a way for chemists to work on an experiment, on experiments that can show us from chemical complex systems some lifelike signs, if you wish. So this is one place, as you see, on the knee of the River Rhone, the University of Lyon, and I'm working in a lab which calls itself Systems Chemistry of Biomolecules, and we are very much guided by the thought that Richard Feynman, actually shortly before he died, wrote on his blackboard, "What I cannot create, I do not understand." Of course, he was a physicist, but I think for us chemists, it's a very important message that he wrote on this blackboard. So the title of today's talk is Preparing for Self-Evolving Synthetic Cells. Preparing means work under construction, yes. The importance of translation in evolving mixed lipid compartments. Translation in the sense of, not in the sense of Stefan Mathil's talk, physical translation, moving, but rather, so in French it would be traduction, yeah, the translation from one language in the other, the more metaphorical meaning of translation. And so before I dive into our experiments towards the end of the talk, I need to clarify first certain points. For example, I'd like to speak a little bit about systems chemistry, about the essence of systems chemistry, about what is systems chemistry of biomolecules, why systems chemistry of biomolecules. I think I need to go back and resume a little bit, recall how it started and what were the main questions that were asked when systems chemistry started, I don't know, 10 or 15 years ago. And then I'll get to a discussion on low digit and high digit polymers, which is quite theoretical, but I'm sure many of us will immediately, connect to it because it's the audience that realizes molecules of this kind. And only then I'll get what I call to the real thing, meaning our experiments, which will be much less designed than I used to work until, say, 5 years ago. So, systems chemistry is something which was coined quite some time ago, maybe 2005. And in the meantime, we have the Journal of Systems Chemistry at Wiley, and some of our speakers actually yesterday published or showed work that was published in the Journal of Systems Chemistry. And it's a very wide area. I used to describe it as something very general, like in this cartoon where you see a chemical reaction realized, an ester hydrolysis, and this ester hydrolysis has gives some response which makes a network to get making more of it. So this is a little essay that I wrote in Life, The Essence of Systems Chemistry, and I'll not dwell too long on that. I just want to point out the fact that systems chemistry is less about single molecules than about how they interconnect and how they get into making networks. And so more of it does not mean necessarily more molecules. It can, of course, but it can also mean more of some signal, more of an intensity, more of a clear network out of the background, and so on. So this is described in this paper, and I'll switch quickly into why systems chemistry of biomolecules. Well, when you look at the bricks of the living nature, I mean the bricks, the large bricks, you see these wonderful secondary structures and then tertiary structures or complicated supramolecular organizations of 4 biomolecular classes, actually 3.5, I would say that, because nucleic acids are carbohydrates as well. So for example, phospholipids are amphiphiles, and amphiphiles form spontaneously compartments. These amphiphiles can form spontaneously compartments. And they can replicate their shape. Yeah, it's not an information. It's also some kind of chemical information. Spontaneously replicate shape. Spontaneously make more vesicles out of few vesicles when properly fed under good conditions and so on. Then carbohydrates, of course, hydrophiles. Yes, usually. I mean, cellulose is not water soluble, but the smaller ones, yes, they serve in living nature as the larger ones as external identification, antennae, yeah, on the surface of cells, and the smaller ones are mainly food supply, I would say. I hope I don't hurt any carbohydrate chemist in this audience. Proteins, I don't have to say too much about it. Of course, they are the all-rounders. They know how to do actually anything, really, if they properly evolved, yes, or self-evolved in the living nature. So they are the perfect catalysts and structurants as well. They structure the matter from which has become alive. Last but not least, of course, there are nucleic acids. And nucleic acids, they serve as an internal identification, but they also serve parts of it, the germline DNA, as a systemic, in larger organisms, as a systemic memory. And so you see these white, you see the white arrows. The white arrows indicate the network, and the network is not complete, of course. The proteins, they do actually the main job. They produce the phospholipids, they produce the carbohydrates, they produce the metabolites for this. And it is true that nucleic acids themselves also can directly produce some of the stuff. I mean, ribozymes are known, but the activity and the importance, the direct production of something is much less than that of proteins. However, there is a very important interaction, a mutual synthesis, a mutual dependence of two classes of compounds in living organisms between nucleic acids and proteins. Proteins make nucleic acids make proteins, yes, and one is a polymer, the other one is also a polymer. One is a quit polymer, a 4-letter polymer, the other one is a viginted polymer, a 20-letter polymer. So the core of living nature, of a chemical system that is living, so it's a biological system, is actually the way the quit and the viginated talk. So the genetic code in the translation, traduction, yeah. That's the systems chemistry of biomolecules. And if you want to understand it in the sense of Richard Feynman, then actually we need to somehow understand how any genetic code can originate. And that's, of course, not an easy question, and even more difficult to answer. How did this systems chemistry start? I mean, I'll not talk too much about what life means, but we can maybe argue that one important thing is the persistence of thermodynamically not so stable matter through reproduction. So the physical explanation is it's matter that reproduces, and that way, through generation, persists, although the individuals degrade normally through the second law of thermodynamics. Biologists would say this persistence comes about from the replication of genetic information, which does the whole then the rest. Chemists would say, okay, that's persistence from the replication of linear macromolecules. And so, for example, I mean, one of the most important influences on this point was Günther von Kiedrowski from Bochum, who published very early abiotic templated ligation of oligonucleotides and could make them self-replicate. So he of course had some Nature papers in the 90s, but the first paper that showed this was in 1986, where he synthesized synthetic DNA blocks A and B, and the A molecule is activated selectively by a condensing agent that is very well known. You see here the group that is already sitting on the phosphorus, on the 3' phosphorus. The 5' end was blocked on the B partner molecule. The OH group was the nucleophile, and the 3' end was blocked. So elongation was blocked, and only ligation was made possible, enzyme-free, of course. So two solutions, you mix them, and they produce a product, which itself is the template, and thus autocatalytic. And this was an enormous discovery at the time. It took some time until it started to grow into the community, and we call it amplification and replication of oligonucleotides. And here you see the kinetic scheme of this paper. And what Günther at the time did was measuring by HPLC the time dependence of all oligonucleotides that he had in the solution, and he discovered something which became known as the square root law. So it is about how making more of it, and the it in this case is T, or TT if you want paired. So the rate of this reaction, the initial rate, without saturating effects by product inhibition in the end, so it was or is dependent from the square root of the of the product TT, which means, in a differential way of describing, is that this rate is slowing, the rate is slowing down, actually, yes. And if you integrate this, you get a growing concentration of the template of the product. It can be a density of whatever, but the way it is growing is called parabolic growth rate. And now when you think of living systems that replicate, then this differential form is linear. It's a constant rate and constant rate acceleration, a constant differential rate, and it integrates into an exponential growth rate. It also worked on peptides. Reza Ghadiri from the Scripps Institute, he was fascinated by the work of Günther and showed about 10 years later that you can do the same thing with peptides. So here you see two red and blue peptides that can ligate to a gray peptide, and the gray peptide is the product which is autocatalytic in its own formation, and what was in Günther's case the base pairing was here the hydrophobic interaction between leucines and valines designed to be on one side along a helix, yeah, and the two ligation the two bits that to be ligated were designed as being a nucleophile, a thiol over cysteine, and the benzyl thioester, which was the electrophile. The chemistry, the molecules, everything completely different. Yeah, the ligation, for example, was a native chemical ligation reaction, gave the same square root law. This was fascinating. This meant that this is not dependent on really the molecular class, and maybe still other classes, completely different molecules can, if designed properly, can behave that way. Yeah, make more of it. So let's talk just a little bit about the population growth kinetics of these growth regimes. So you have some density or a concentration of some population, molecular, cellular, whatever. Yeah, you can take elephants actually, or people. At a time 0, time and other times, you have a population growth rate constant, the rate constant that we know, K. And then you have P in the exponent as the growth order. So if P equals 1, we can call it it gives the exponential growth. We can call it forceless because, well, I'll show you just in a minute. Then if P is smaller than 1, then you get into this inhibited or parabolic growth rate, for example, square root, but it just needs to be lower than 1, the P. Of course, if P is higher than 1, for example, 2, then you can call it hyperbolic or accelerated growth order. It's easy to show in the formula that for the forceless growth order, you get time in the exponent, which means these are simple replicators. Forceless because the formula is formally the same as Newton's force formula. Simple replicators in every generation produces the same number on the average, of course, of descendants per parent as the previous one. Yeah. However, for example, integrate this, and you see it can be integrated easily with a square root, luckily. You see that T is not an exponent. This parabolic growth means every generation produces fewer descendants on the average per parent than the previous one per parent. Yeah? And of course, if you take, for example, P equals 2, then you get this formula for a hyperbole, and this means every generation produces more descendants per parent than the previous one. So, for example, the human race, yeah, or chicken, or I don't know, tomatoes, cows, they're all linked to humans somehow. We are in the hyperbolic growth regime. Wild animals, that are not particularly, you know, molecules, for example, PCR, yeah, bacteria in some medium, or PCR reactions in vitro. They grow in the forceless growth regime, and molecules like self-replicating molecules like the one that I showed are in the parabolic growth regime. So there was Urs Satmari from Budapest who realized that this is a huge problem for the start of it. How do you get living? How do you become alive from a mixture of a chemical mixture of stuff? Yeah, because the survival of the fittest, meaning the Darwinian evolution, which you know, the mechanism of it is mutation selection of the fast replication population. it operates only in the exponential regime, or at least in the exponential regime. And it was him who realized it's mathematically actually not so difficult to show that when you have a population of different parabolic replicators that have different k's but the same p or below 1, then there is no way how they can outcompete one another through faster replication. There is a, he called it survival of everyone, which is of course a nice thing once you're living. But before you're living and you're just a chemical mixture, how do you, the speciation, yeah, how do you get out something which can be defined as something that will persist and you know, have an identity in whatever way? So this was a big problem, yeah. The problem is that parabolic replicators, they grow through a mechanism, a templating mechanism, as DNA and RNA in living beings. However, there is this thing that strand inhibition. So DNA, double strands, RNA, RNA, peptide, if well designed, the templating thing that makes it autocatalytic at the same time blocks full free access to the single strands. Either you stick to or you don't, but or half. But then the more you stick to one another as strands, the more is scavenged in the double strand, which makes it slow down and more parabolic. So without enzymes that keep the single strands apart, you will never get into exponential growth. So it was Urs Satmari also who found the solution to this problem. The solution was that if you compartment parabolic replicators, so these small dark and rings, filled and empty rings, these are the molecules that I was talking about that replicate in the parabolic growth regime. If you compartment them in something that in vesicles, which are shown here as the large circles, and you can make them these divides. So from one large circle, it grows and then it splits and makes 2 or 3 more, and their content will be stochastically distributed, and the fitness of these protocells, or you know, these compartments, will be connected actually automatically to their content, meaning that not all contents can survive or not survive, but the vesicles, they can. Some can be fitter and the other one won't, and that's the way how maybe it could get started during the origin of life sometime. So I think, in my opinion, this is the most fundamental reason why life needs to be cellular. Apart from the fact that we protect, that we concentrate, all the reasons that are given, that are immediately seen by chemists, this is one fundamentally important reason. There is another problem, the problem number 2, I call it. Replication fidelity limits polymer length. There I have to go into a little bit a discussion of the information content of linear polymers. You can define as Shannon information the maximum information that can be transferred from a string of digits into other strings of digits. Yes, and it depends whether so the digits can be binary, ternary, quaternary, or more. And the n is here the number of the length of the strings, or the number of the residues in strings. So imagine a bit polymer, yeah? So you have just zeros and ones, like G and C only, for example, in a DNA. And I took out a 40mer just for the fun of it. B is 2. The information maximally, the maximum information stored in such a bit string is 40 bits, yeah? And it can be doubled, and then can be doubled again and doubled again when using the complement copy rules. Now take a trit string of the same length, 40, B is 3. The information content is higher, according to the maximal information length is higher than according to this formula. It's clear the information density is higher. And so there are copy rules. A trit string needs at least one self-complementary recognition. I think for chemists, it's maybe not so difficult to see that it's rather difficult to be strictly self-complementary than not self-complementary strictly. Yeah. nature, biological nature, so the biological memory polymer, the DNA and RNA, the nucleic acids that we know, these are quit polymers, 0, 1, 2, and 3, for example. So you take this length and you see with B40, these are 40 quits, okay, but these are, the content of information in bits is 80, doubled. And the copying rules, of course, are very well known. The copying rules are the famous Watson-Crick rules. So you take 0 for adenylate, 1 for guanylate, 2 for uridylate, and 3 for cytidylate, and you get the Watson-Crick pairs that are the most stable ones. And we know, of course, that molecules do whatever they do, what is possible. So there is very well known are the wobble pairs, of course. We also know the structure and the possibility and the stability of Watson-Crick-like pairs, the 2 short and 2 long ones. And there are other geometries, like, for example, Hookstein. So these are mismatches and they are less stable. So it all boils down to the match versus mismatch stability and incorporation speed, yeah? The efficiency of forming matches versus mismatches, which translates into this sentence replication fidelity limits maximum polymer length, which was first described a very long time ago. I mean, in the 70s, 1970, 71, 72, 78, and so by Manfred Eigen in the Max Planck Institute of Göttingen. I'll not get into the maths of that, of the error threshold. I just want to pick out one thing that has become the name of Eigen's paradox, which is there is no accurate replicase without a large genome, whatever large means, and there could be no large genome without the accurate replicase. It's a chicken and egg problem, yeah? Thus, the information that can be reliably replicated is less than the information necessary to code for the replication machinery. There is a way to solve this problem if you allow for translation, traduction. If you allow the low digit polymer, to parts of it at least, to translate into a high digit polymer. So here you have a 27-mer picked out of this 40-mer. You can have 3 frames, yeah, and through some code, for example, I just wrote the biological code, which is a triplet code, produces a gene product, which is then the protein, of course. That's a gene product, which in principle has a digit number of 64, which is then reduced to 20 in our nature that we know. The information, the maximum information content is the same. Yeah, let's think about that, dwell on that a little bit. B4 means that's a low digit number and a low diversity. Now, I'm not sure whether Dipankar, for example, is listening, or people who are convinced that you can make cells with RNA only, but the catalytic potential of nucleic acids is not very large. That's my conviction. However, the low number of digits means they are easy to distinguish. It's easy to distinguish between 0, 1, 2, and 3. So the mutability of mu D means the mutability of each monomer is held low. Yeah. So it is easy, relatively easy to copy such strings, and that's why they can be inheritable. However, when you produce a high digit polymer, then the diversity becomes enormous, and with it, I'm convinced, and one would have to show that in a more stringent way, really, that the higher how much higher the catalytic potential of these materials are. The digits B prime of these highly diverse gene products, they are difficult to distinguish from one another. I mean, distinguish K from L or M from a big M, or, you know, a dollar from a euro or whatever. So they cannot really serve as a faithful copier. Yeah. So they have to be resynthesized from the low digit every time in each generation. However, they are such good catalysts that they can help overcome the low digit polymers, the parabolic growth regime. And this, of course, you know, makes it livable. I don't know the English word for it, really. So in conclusion, for useful information to be selectable and stable over generation, you just need dynamic compartments to have early, and you need efficient catalysts like replicases and helicases, you know, to keep the single strands apart. They must appear early during the evolution. So in other words, no life without lipids and no life without translation. You can also, you know, you don't have to stick to DNA and peptides. One, as a chemist, one could design other systems of bit strings that are translated into high bit polymers. They could be through doublets, through triplets, to quadruplet codons, through more. It is you can take trit strings. You don't need to have a DNA or RNA. As I said, you can take quit strings and use other than a triplet codon. Anything is possible. What I want to say is that translation, the fact that you have low digit and high digit, is fundamental, together with the cellularity of it, and that translation does not really mean from one class, not necessarily just mean translate from one compound class into another, but from a low digit into a high digit. So I was thinking about, for example, this morning's talk that described Nikos Hatzatzki's and Stefan Vogel's work. I'm not sure whether, you know, the single particle combinatorial liposome fusion mediated by DNA. Fascinating. This, I'm not sure if this is actually already some kind of translation, just that the gene product there is not really synthesized automatically from the genes, and it's supramolecular. So what I'm thinking about, the importance in chemistry would be to find systems where low digit polymers produce covalent high digit polymers. And this biological network boiled down to just a general way of saying low digit catalyze catalyze the high digit formation. However, the high digit polymers do catalyze the exponential replication of the low digit polymers, and they catalyze the translation as well. So actually, a ribosome, for example, is the ribozyme. There are nucleic acid polymerases that are proteins, and very important is the amino acyl tRNA synthetases. Without those, without this correct recognition of the tRNA and the amino acid, the genetic code doesn't mean anything. Yeah, and these are proteins that know how to recognize amino acids from. So there are from one another and recognize their cognate tRNAs. So there is I just want to say that there is Charlie Carter from Chapel Hill and Peter Wills from Auckland, who are heavily working on this aspect. I think this is very important. And now to the real thing. So what are we having in plan? We have a plan of a chemical system that we would like to see to show us somehow a lifelike behavior. So one sub-plan is, one project is to make giant vesicles reproduce. We produce them from gentle hydration of thin lipid films. Yes, this doesn't give us unilamellar, everything. There are very huge mixtures of quite large compartments, which is good because we want to use selection for our experiments. We will not design, we will want to give a population of different things. We want to feed them with what we call incomplete lipids, not phosphorylated lipids, make them grow in size and divide, so grow the population, and then somehow add what I would like to call here geochemical anhydrides, which would then phosphorylate these lipids and make them more rigid and more fit for the next generation. That's one sub-project. The other one is the internalization of RNA and the evolution of compartmented molecules. So you see here below, lipophilic oligopeptide libraries, a mixture of lipophilic peptides. It will be brought in contact, or is brought in contact with internally labeled oligonucleotides. This mixture, with the help of condensing agents, produces amphiphilic oligopeptide oligonucleotide conjugates, which themselves will be, brought into contact with giant vesicles that they contain inside a clickable fluorescent dye, which cannot leak out. And so when we incubate these in a certain rhythm, then we should compartment some of the oligonucleotides, and we could just show this by a click fluorescent dye. Yeah. So this is quite a large project, and my group is not so large. So we have, for example, Dimitri, who worked a lot in his PhD on oligopeptide, oligonucleotide conjugates. I will not talk too much about that. We have Augustin, who worked a lot in his PhD on the growth and division, and I'll present you some work of this. We have Carolina, who is a hard worker, and she is producing clickable fluorescent dyes. And we have Anastasia, who is trying to figure out geochemical anhydrides that could be useful for our purpose. And now this, you see, this will have to close into, will enlarge into the project where we add these molecules and we feed them mixtures, and we feed them also with lipid amphiphiles at different temperatures. We make them grow and divide, and we see what happens with the molecules inside and what happens with the membranes. Yeah, and we have different various methods about ideas how we can analyze, so we can get ideas whether the amino acids and the peptides influence the RNA or the DNA or the mixtures, and whether we can see something. And somehow you have to bring into contact amino acids and nucleic acids to be able to get an experimental idea of translation, at least of peptide growth. So the work that I'd like to present to you a little bit in more detail is something that we just published. It's the chemical analysis of lipid boundaries after consecutive growth and division of supported giant vesicles. The main author actually is Michele. I'll present to you Michele at the end of the talk. So this is a graphical talk. We have vesicles that that inside have a glass bead, yeah, and we feed them with stuff, and then they grow the membranes, and they detach the parts of the membranes and put them into the solution, and we can do this several times. The idea is based on a technique that Michele has developed to use glass beads. My glass microspheres, absolutely all are 5 micrometers. We cover them with avidin, and then with a lipid that contains biotin. So we anchor this biotin lipids, and then we can, add very different phospholipids, mixtures of phospholipids, pure phospholipids, what have you. Also fluorescent phospholipids, some of it, to make these supported vesicles, yes, the glass supported vesicles. The number of the biotinated lipids is, let's say, the number of all lipids on such a membrane is at least 2,000-fold higher than the number of the biotinated lipids. So we have this technique, and here you see in the structure the lipids that we use: POPC, POPA, DOPC, DOPA. You see also the biotinated structure and the rhodamine. And what we feed with are the feeding amphiphiles below. So oleic acid 7, yellow; myristic acid 8, green; and myristoleic acid 9, red, palmitic precipitates. Here is the experiment. It's a consecutive addition of sorry. This is a consecutive. I'm going here. Sorry. So here you see these supported vesicles. They are fed, for example, with oleic acid. What happens is that the supported glass beads, they are more heavy, and they are below and above are the free-floating vesicles that form. Yeah, we can separate them through centrifugation. We can analyze the phospholipids we did through the Stewart assay, which is a colorimetric assay on organosoluble organophosphates. We by HPLC, without a chromophore, so with an ELSD detection, we determine the content of the fatty acids separated in the host solution and on the beads on this microsphere, and then feed again with another lipid or with another fatty acids, do the same analysis, and again and again. We can also look at it under the microscope. So this is these are epifluorescence microscope images, fluorescence, but epifluorescence microscope images. The lens is from below, so we can also see the time dependence of it. You know, the first 45 minutes, for example, small vesicles appear and float, and then during hours they become larger and so on, and then they can be separated at earlier time points and at later time points. We also observed some of it under the confocal microscope, so we could really show the detachment or see the detachment, not in a film though, if somebody wants to ask, but at different places, different situations, which are consistent with the movements. And so we can say, yeah, it's possible to have a model system which gives us an idea of actually what happens with the lipid molecules in the membranes during a growth and division procedure. Yeah, and not only that, we quantify it, and so we get the idea which fatty acids have which properties with respect to this growth and division process. And we did it several times. You see here experiments on a pure on one single phospholipid that is fed twice or thrice, or a mixture of 2 phospholipids that is fed twice, or a mixture of 4 phospholipids, quaternary mixtures that is fed twice with 2 different, or then the order of feeding also, which gave us an idea what to do best to be favorable for the experiment that I described to you that we have in plan. Yeah, the data is quite clear. One conclusion is that saturated fatty acids stay more. I mean, the problem is that you don't want to form vesicles directly. You want to feed them into the membranes that you already have, so you can keep the content. So with that, I thank the collaborators, Michele Fiore, Dimitri Fayol, Augustin Lopez, Carolina Chieffo, Anastasia Shvetsova. I also thank Isabelle Daniel, who is a PI in the geology department. I'm just showing you that it's Michele and not Michel. I hope you clearly see this. My group is wonderful. We work a lot, and you see before the COVID era, and then Anastasia joined us in 2020. It's easy to see. Isabelle Daniel, actually I'm co-supervising Anastasia in her PhD project. The ideas, the way we are thinking and how we are working is profoundly influenced by cost actions, the 3 cost actions that I had the privilege to be part of it. So I'd like to thank the Cost organization, also the European Science Foundation. I very much thank the Volkswagen Stiftung, who the real money for the experiments. We got a project on a call called Life, a fresh approach to the basic principle of life. Our project is called Molecular Life. So I thank the Volkswagen Stiftung very much, and I'm sorry to say I don't thank at all the Agence Nationale de Recherche, who just never funded any of this research. And maybe this will change. This will be beautiful. And I thank you for your attention, and I'm glad if I can answer at least one question. I don't know how the time is.
Great. Peter, thank you very much. It was a great talk, and you gave us a lot to think about. We do probably just have time for 1 or 2 questions, and it would be wonderful to raise your hand or speak up.
Philippe.
I see a couple Philippes, it looks like.
Yeah, thanks, Mark. Thanks for Peter, for this very nice presentation on very fundamental aspects, and thank you for the introduction on system chemistry. So I have several comments, and the first one is I was wondering if the system chemistry can be seen as a dynamic supramolecular chemistry. In fact, we are looking at the all those molecules moving together and interacting together. So I was wondering if we could see the system chemistry as a sort of dynamic supramolecular chemistry. So that's the first comment. And the second one is, we know that all those molecules are interacting together, but they are all organic molecules. And I guess inorganic molecules are also playing a role in the translation of the information or the replication process, at least at the early time of life. So I would like to have your comments on those two aspects.
Thanks, Philip. Yes, when I teach about systems chemistry, I usually say that it's like supramolecular chemistry, dynamic supramolecular chemistry. It's one step, meta step on above or embracing supramolecular chemistry. I agree very much with this way of seeing systems chemistry. Yes, inorganic material are extremely important. For example, clays, minerals. I mean, I didn't talk about it, but the geological part, you can imagine, will not be organic. They provide surfaces. This is part of the most important catalysts are metals and minerals. So absolutely, you cannot just confine the systems chemistry into organic molecules.
Thank you. I think we have time for one more question as well, please.
Okay, thanks. Thank you for the very inspiring talk. I'm Philip, that is working with some lipidated nucleic acids in the Stefan Vogel group. I was very impressed by the big conjugates that are able to transport nucleic acids through the membrane, and this is quite intriguing, I think. Have you thought about what could be harvested as what could be encoded in this nucleic acids? Right now, you're thinking about maybe ligation amplification, or you're thinking about actually the peptides having a catalytic function, or so on. I would be very happy to discuss this.
Yeah, maybe the symbols suggested two long nucleotides, yes? To get through the membrane, I'm convinced that you cannot be too long. I mean, it very much depends on actually the membrane composition, I'm sure, as well. But I don't think that sufficient coding can cross the membrane. I think that inside, you have to somehow get in the RNA or nucleic acids. Other people would say, why do you have to use peptides? Well, because peptides and RNA it's a primordial interaction, which should be tested for several reasons. However, to become a length that can code something useful, I think this would happen inside. So,
yes,
so the chain length inside is very important. It's just a start to get them through.
It would just be very interesting how chemistry on the inside could affect, for example, processes like the division of cells could both maybe reinforce the membrane or destabilize the membrane or change the membrane's properties towards, for example, binding inorganic materials and so on. So I think it's very, very cool that you have thinking about feeding a system from the outside, but actually, yeah, going all the way to what's going on the inside. So
thank you.
Great, thank you very much. I think because of time, we'll move on, but by all means, reach out to Peter via text or email. I'm sure he'd be glad to answer and discuss. So again, thank you very much, Peter, for the talk.